Hi, my name is Annie. Today I'm here to talk to you about my project titled Hints of Novel Immune Signaling Pathways. Eukaryotic serums are capable of NAD degradation. Now, that's a lot of words. And to be honest, when I first joined the Morehouse Lab, I did not know what a single one of them meant. In fact, I was freshly 18, and not only was I struggling to adjust lab work, I was still figuring out how to be an adult. I wore mismatched socks every day, always forgot to pack lunch, and I once had to bail on an experiment because I threw up due to food poisoning. At that point, I really had no idea how similar I was to that little bacterium ransacking my bowels. Bacteria are constantly under threat of infection from viral bacteriophages and have evolved a diverse arsenal of defense systems to protect themselves. One of these defense systems is called the Thor's defense system, where, upon pathogen detection, a bacterial protein called ThisA will cleave the cellular metabolite NAD using a serum domain and execute an immune response. The human innate immune protein, Sirims, also contains a serum domain and is able to cleave NAD. It is hypothesized that Sirims is evolutionarily derived from the ancient prokaryotic serum domain inherited from the endosomatic event that gave rise to the first eukaryotic cell. If that's true, we would expect to find NAD cleaving serum enzymes in diverse eukaryotic organisms. To investigate this possibility, our collaborators employed a homology-based search to find other possible eukaryotic serums. Their work resulted in 9 possible eukaryotic proteins for me to test for NADase activity. To do so, I purified these proteins and incubated them in a test tube with NAD. To observe for NAD cleavage, I separated the reaction products using high-performance liquid chromatography. Out of the 9 possible serums we selected, we identified 2 serums that were able to cleave NAD, one from zebrafish and one from plankton. Here, we observed that these serums are able to split the molecule NAD into its 2 constituent molecules, ADP ribose and nicotinamide. But Annie, you might say, why should we care? Well, first of all, NAD cleavage hasn't been implicated in other immune signaling pathways, such as with the human trypsin-2 calcium ion channel or in plant immunity, suggesting that these new serums may be involved in previously uncharacterized immune signaling pathways. Secondly, zebrafish and plankton are from distinct sections of the eukaryotic domain, yet they both possess similar NADase activity. This strongly points to the presence of serum in an early eukaryotic common ancestor, and is not just a product of multiple de novo developments. And most importantly, just as immunity is ever evolving, I too am always growing. As an undergrad of the Morehouse Lab, I have learned and experienced more than anything I could have ever imagined. Indeed, I sashay every day to work with matching socks, only occasionally heat up my packed lunches in metal bowls, and have ceased to DoorDash suspicious takeout before experiments. So, to the Morehouse Lab and our collaborators for helping me grow, thank you.